Hi, in this video we'd like to continue discussing enzymes. In particular, we want to focus on the mechanisms whereby an enzyme will reduce the activation energy of a reaction, and then we'll end up talking a little bit about protein folding. In this first slide we see in blue an enzyme called lysozyme, which acts upon polysaccharides, which are strings of sugars, which are shown in red. The reaction cleaves a longer polysaccharide into two smaller fragments. And here you can see some of the detail. You have a glycosidic bond between two sugar molecules. You have two amino acids, glutamic acid and aspartic acid, making simultaneous attacks on the glycosidic bond, proceeding to a covalent intermediate with this sugar. And then we have hydrolysis of a water molecule, adding a proton to the glutamic acid and putting a hydroxyl group on the carbon, cleaving this ester bond, which is an intermediate. And that's pretty nice, but I feel that's still fairly complicated. And so I'd like to help build your intuition about enzyme, a kind of enzyme mechanism. And really the only rule you need to know is that opposite charges attract, like charges repel. This figure from the book shows three mechanisms of enzyme catalysis. The first is undeniably important. It's simply an enzyme which will hold reactants in the proper orientation near each other, giving them plenty of time to react and not relying on fleeting interactions, collisions in solution based on diffusion. The second mechanism is one we'll talk about more, and that is electronic stabilization of the transition state. However, this picture is somewhat flawed, as you don't see a transition state. You have a charged molecule. What you see is electronic stabilization of a molecule itself, and we'll talk about that. And finally, I think I disagree with this third mechanism of physically squeezing or bending a molecule, but perhaps I don't understand exactly what they're talking about. So now we'll talk more about mechanism 2, and we'll also use that to discuss some features of protein structure in a little greater detail. Now let's talk a little bit more about an amino acid. In solution, we've talked about it being a zwitterion with a positively charged amino group and a negatively charged carboxyl group. And here is a peptide bond with the C-alpha carbon, another amino group here, and another carboxy end here. If we look along these four atoms in the peptide bond, there's actually a fairly significant dipole moment where we have excess electron density at the oxygen end and relative electron deficiency at the proton. So that results in a dipole moment of about 3.5 dBi, which is quite large. And I should just mention that for whatever reason, who knows, in biology the convention is different from physics. In biology, the arrow points towards the positive end of the dipole. That's opposite what you may have learned in physics class. So now I'm going to talk about what happens to those dipole moments for each of these peptide bonds in the helix. Unfortunately, the orientation is upside down here, but what we have for those hydrogen bonds, if we can look, here is a peptide bond. We have the oxygen, the alpha carbon, the amino group, the hydrogen, and so we have, in this case, a dipole, but is pointing almost along the alpha helix axis, and we can see many of those. Here's another one pointing up. Here's another one. Now in this slide, it's emphasizing all the different dipole moments of the individual peptide bonds or amino acids, however you want to think about it, and these will sum into a fairly significant vector that points down the alpha helix. This has the effect of creating fairly significant electric fields at either end of the alpha helix. Now let's imagine a hypothetical active site where there's going to be a molecule reacting in here, and we have 4 alpha helices lined up pointing towards the active site. It's not at all unusual to have this sort of orientation, although it would be spread out in 3 dimensions rather than in 2. Now, to understand a little bit more about the stabilization of the transition state, we need to think about a molecule and a generic transition state. And I've just drawn a molecule with 5 atoms here, and we're going to pretend that this enzyme is going to cleave between bond 3 and 4. As it goes towards the transition state, so here's the starting material, a normal bond between 3 and 4. Here is the product, a broken bond between 3 and 4. And the transition state is stretching it's starting to stretch bond 3 and 4 longer and longer on its way to being broken. Because you have a certain amount of electron density between atoms 3 and 4, stretching it out over a longer distance means there's going to be less electron density in the transition state relative to the ground state. So you can think of sort of a partial positive charge appearing in this instance at the midpoint of the bond. So now we're going to place the transition state of the molecule in our hypothetical active site. So now we're seeing the dipole moments that sum on the alpha helix from the individual amino acids and peptide bonds, and we see that focused here on the active site where we have a partial positive charges, we have partial negative charges at the ends of all the alpha helices. This is going to stabilize the energy. Otherwise it's again, because opposite charges attract, that means you're lowering the energy of putting a positive charge in this region. This is a way in which you'll reduce the energy of the transition state, leading to an increased reaction rate. Now, you have to remember that neither the starting material or the reaction product has this buildup of or deficit of electron density or buildup of partial positive charge. It's only the transition state that has this positive charge, so it's only the transition state that is stabilized by this orientation of electric fields at the active site. So while it's not essential to answer the typical biology type questions about how enzymes work, I think it's so simple once you break it down that it's provides some insight into how enzymes work in general. I'd like to say just a few quick words about protein folding because it's such a complex subject and not completely understood that I don't want to delve into it anymore. But as we know, that proteins are really only active in their folded forms. In their unfolded forms, as we see here on the left, this is not going to be capable of doing the kinds of biochemistry that we want it to do. On the right here, we can see the folded form with an alpha helix, 2 beta pleats in the final folded form, and now this is the active form. So the question is, how do you get from here to here? And do you think it's a challenging problem or not? Well, it turns out it is a very challenging problem in principle. And Cyrus Leventhal pointed out, in fact, numerically, just how challenging it is. Imagine a protein made up of 100 amino acid residues. And just to start things off simply, let's just assume that there are 3 conformations for each amino acid. That means the total number of conformations for this molecule is 3 to the 100th power, which is an astronomically large number. He then went on to postulate, assuming that it takes 100 picoseconds, so less than a nanosecond, to convert from one conformation to another. It's an unbelievably long time for just a single protein to sample all the different conformations. is the only way that you could rigorously know that you're at the absolute global thermodynamic minimum when you've done folding the protein. Contrast this with what's known about protein folding in vivo, which is that it can take microseconds or milliseconds for an unfolded protein to fold. Clearly, the protein is not sampling all conformations as it's folding itself, and there's some fundamental process going on whereby it can achieve a folded state much more quickly. And this slide encapsulates much of the current thinking about protein folding. Here we have the linear chain, the primary structure, and what you have very rapidly is a collapse of local amino acids into stable structures, alpha helices and beta sheets. So it's thought that secondary structure units form very quickly, and now instead of having to sample conformations for, maybe 12 or 15 amino acids here, this alpha helix can act as a single unit, and we can begin to arrange conformations of just this one unit, which drastically reduces the number of potential conformations available to a protein if we lock in a bunch locally with these secondary structure elements. The next thing that occurs is the cell trying out relative orientations of the various secondary structural units, and when they achieve some stabilization, then you'll get some of these loops that sort of are not recognizable secondary structural folds packing in around those, and then the protein will minimize the energy of conformation settling into a fully folded state. The other important aspect which drives this process is that those hydrophobic residues will quickly try to seek other hydrophobic residues to sort of coalesce around a hydrophobic core, minimizing the contact with the aqueous solvent. An important term that you'll hear in this discussion is which is around this stage, is called the molten globule, where this linear chain has started to condense into roughly a compact form, yet it still has a greater spatial extent, as you can see here, than the final product. So it's compact, but it's still a little larger, so that it has room to move around and sample some conformations between residues. Okay, that's all I wanted to say about protein folding. Thanks for listening.